Thank you, Sandeep. So at the outset, I must thank Professor Umesh, Tanweer, Raj, and Sandeep for organizing such a beautiful conference and giving me the opportunity to present my work on UPF3B protein. So my talk will be divided into two small sections. One section will be introduction, where I will introduce NMD process, which is the most important process for leading into RNA decay. And in the next small section, I will talk data from my lab, especially on the knockout of UPF3B and the inferences we take out from these experiments. So let's start. So as you all know, when a normal transcripts are translated, it gives rise to full-length protein. And these kind of transcripts are not rapidly degraded, right? Whereas in terms of premature translation termination codon, this transcripts actually has a premature termination codon, which disturbs the open reading frame, leading into truncated protein if translated. And this kind of transcripts are rapidly degraded in the cells. So that means there are signatures, and this NMD process is tightly linked with translation dependent process, as well as this translation dependent process has the potential to discriminate between normal termination codon versus premature termination codon. So let's look into the translation dependent termination processes. For example, after a translation starts, during the elongation phase, we all know that tRNA, amino acyl tRNA, is brought about and joined the ribosome with the help of eukaryotic elongation factor 1. Now, upon the entry of stop codon, this stop codon can be recognized or decoded by either release factor 1 and release factor 3. Now, depending on the interaction partners of release factor 3, this GTPase bound is actually displaced, and the end of this GTP is hydrolyzed, and release factor 3 is dissociated from this position. Now, exactly at this time, you have GGQ positioning exactly at the center, and this GGQ positioning helps in the hydrolysis of a tRNA bound peptide, which is further actually stabilized by recycling factor ABCE1. And this is exactly the position where earlier release factor 3 was bound. So making it the whole process into the recycling phase, the ATP bound then allows the whole ribosome to be splitted with the help of ATP, and the splitting is done. Here, release factor 1 is responsible for releasing peptide chain. Now, this whole process reinitiates. Now, sometime during the termination codon, there is a distinction or decision has to be made whether the stop codon is either normal or it is actually a aberrant stop codon. And this decision making is done with the help of release factor 3. And depending on the binding factors of release factor 3, the termination is either considered as a normal, which is not degraded faster, whereas if it is aberrant, then it is rapidly degraded. Now, as I've said, if you consider this as a normal transcript, what are the features in normal transcript? You have a stop codon, and if you have a normal 3' UTR length, then poly A binding protein is in the proximity of the normal stop codon. So upon translation, this poly A binding protein stimulates this termination process, and therefore there is no recruitment of NMD factors, and there is no degradation or rapid degradation, I must say. Now, in terms of PTC containing transcripts, we have PTC, where the ribosome generally stalls, and this makes a larger distance between poly A binding protein. Therefore, this protein is not able to stimulate the normal termination codon. Hence, NMD factors are recruited at this position. Having said that, There are another factor, for example, here, exon junction complex. I know some of you are saying that these are old, but still last month, everything, all NMD biotypes, transcripts were actually based on EJC 50 nucleotide base pair, distinction between the EJC complexes or NMD biotypes. Now, this EJC factors are deposited along the splicing processes at around 24 nucleotide upstream of exon-exon junction. Now, during normal translation process, all these EJC factors are displaced, and therefore, there is a normal termination. Therefore, there is no NMD factors that are recruited at this position. Now, in case of PTC containing transcripts, what we have is PTC at this position, which removes the earlier exon junction complex. The stallation of ribosome at this position at PTC does not lead into the removal of downstream exon junction complex. Now, this downstream exon junction complex tries to recruit NMD factors, as well as the distance here is also large between the poly-A binding protein and the stalled ribosome. Now, if we see these factors, then a normal transcript without any exon junction binding proteins and with a normal length of 3' UTR, they are NMD resistant kind of transcripts. Whereas due to various errors in the system, for example, splicing error or transcriptional error, irrelevant positioning of EJC in the context of stop codon or premature termination codon can lead into NMD process, and these are different features of NMD transcripts. For example, you have PTC, downstream of that you have exon junction complex. Also, the length here is long compared to the normal transcript. Many times in the 3' UTR, you have alternative splicing condition due to which we have deposition of exon junction complex even at the 3' UTR. Then we already know about upstream open ring frame, which leads into stop codon, and unremoval of exon junction complex again leads into the transcripts which are NMD favored. Now, another kind of stop codon, which is UGA, normally in the presence of selenium, it leads into selenocysteine, whereas in the absence of selenium, it leads into the stop codon, and this transcript is also considered as a NMD feature. Now, various type of physiological transcripts containing long 3' UTRs are also degraded with the help of NMD because of having long 3' UTR. These UTRs could be more than 1,000 nucleotides longer in this case. So, having all these features together and the knowledge about EJC and the poly-A binding protein, let's see how it comes together. Sorry, before that, I forgot to mention about the normal NMD factors. So we have up frame shift factors, which are the core of NMD proteins. Now, these are different types, UPF1, UPF2, UPF3A and 3B in humans. These factors were initially recognized or isolated, discovered in Cerevisiae complex, and later on, homology search revealed that it is also present in higher eukaryotes. If you see UPF1, it is a really long protein. It is one of the most important protein in terms of NMD because this is the core protein of NMD. It is a helicase containing REC-A and REC-2 domain. At a C terminus and N terminus, it gets phosphorylated by another factor of NMD, that is SMARC1. I will come to that later. Now, the role of UPF1 is to compete. It also competes with the release factor 3, and it binds with the release factor 3 and poly-A binding protein. Actually, they both bind for release factor 3. Apart from that, if you see the role of UPF2, now UPF2 is considered also to increase the phosphorylation activity of UPF1. Apart from that, it is acting as a scaffold protein because it has at its C terminus UPF1 binding domain through which it binds to UPF1, as well as through its MIF4G3 domain, it binds to the UPF3B protein through its MML domain. So the role of UPF2 is also known to activate UPF1, and it also include or activate NMD process in this case. When we come to UPF3B protein, its role in NMD is actually controversial. I will come to this. Some people actually say it is necessary for NMD, and some groups actually say it is not entirely relevant for NMD processes because it has another paralog, UPF3A. However, in this case, this is acting as a bridge between two proteins. One is EJC binding motif. At its C terminus, you see this motif, which actually binds to all EJC proteins, namely EI4A3, MAGO, and Y14. And through its N terminus RRM domain, it binds to UPF2. So it acts as a bridge. Now coming to another factor, SMART factors, which was originally discovered in C. elegans, and this both UPF1 and SMART factors are present in eukaryotic system or our mammalian system. Now if you see, SMART is a kinase. Usually its kinase activity is hindered, but when active, that it actually phosphorylates UPF1 protein both at its C terminus as well as its N terminus domain. Now SMARC5 and SMARC7, they work as a heterodimer, and the role of SMARC5 and SMARC7 is to bind UPF1 in a phosphorylation dependent manner. Now the main degradation of RNA happens through SMARC6 because it contains the active endonuclease domain at its C terminus called PIN domain. Now compiling all this together, again let's see how NMD is activated, mainly in the context of EJC dependent NMD process. So for an EJC dependent NMD process to initiate, we have to see that ribosome stalls at this premature termination codon, which leads into the recruitment of release factor 1 and 3. This release factor 1 and 3 actually binds with UPF1, which competes with poly-A binding protein. Because of the distance again, this UPF1, and it has been shown in a literature paper that the scanning model actually brings the concentration of UPF1 close to the stalling ribosomes. Now, this UPF1, because it has a binding domain for SMARC1 and UPF2, it tries to interact with SMARC1 and SMARC2. And more so, because EJC is present downstream of stalled ribosome, this EJC bound with UPF1 tries to interact or bring UPF2 concentration along with SMARC1 concentration. Now, this active SMARC1, the regulation of SMARC1 is regulated by SMARC8 and 9 actually, and this SMARC1 phosphorylates UPF1 both at the N and C terminus, due to which you have recruitment of heterodimer SMARC5, SMARC7 at the N terminus, and at the C terminus you have SMARC6 containing PIN domain. Through this interaction, CCR0 actually starts deadenylation from this terminus, and after endonucleotic cleavage, you have XRN1 and other endonucleotic activity going. So this is in the nutshell all about NMD and how it processes. And there are different bifurcation of this process because UPF1 independent NMD is known. Also, UPF3B independent NMD is known. Now, there are AKT methods which can actually phosphorylate UPF1. So for all this NMD processes, UPF1 is considered as a central factor which initiate nonsense mediated decay process. Yes. Yes.
So do you think
It is even in yeast. Actually, UPF1 is not the central NMD factor, because if you delete UPF1, it doesn't hamper NMD. But that is a completely different system. Maybe the components are different, but I have to look. Not sure. It can be that process, yes. Yes, it can be that. But for eukaryotic system, this is pretty known, because you can get rid of all the components, but as soon as you get rid of UPF1, it is completely lethal. And the endonuclease activity is basically by SMARC6. And in fact, SMARC5 has a pin domain, but that is not active because of the mutations within the pin domain, triad domain. Now, coming to our question, we wanted to actually look what happens in terms of NMD when we knock out UPF3B. So we wanted to understand what is the role of UPF3B in NMD and why NMD. There are various reasons for it. The first reason being when we see patients, a lot of UPF3B mutations were found in XLID patients with the different phenotypes. And with all this mutation, what you see, there is no production of UPF3B. In fact, these all patients have a consistent level of its paralog UPF3A. And despite that, you get this phenotype, meaning that UPF-A is not able to rescue these phenotypes. Now, the second important factor was when we started this whole project in 2018, there was contrasting reports. Some said it is not at all essential. Some said it is really needed for NMD. So we wanted also to see whether what is the condition, at least in our experimental setting. Now, the third important thing that, again, by 2017, it was shown that even UPF3B, at least in vitro, can bind with release factor 3 and can detect all PTC containing transcript for initiation of its degradation. So that was one of the, again, important factor for us to look into this deeper. Now, nobody knows till now why actually UPF3B in neuronal cells are important, because it has been seen that this role of UPF3B in neuronal differentiation. So these are all areas where we want to actually work on, and some of the work we are doing in this area. Now, to know the role of UPF3B, we started actually trying to make stable cell lines where you can inducibly express Cas9 protein. So for this, we cloned Cas9 in a piggyback system, and we transfected it into the HEC293 cells. Here, our inducible system is dependent on the QMAT. And here we have a flag tag upstream of Cas9 and a GFP protein downstream of your our open reading frame. Now, depending on the inducible system QMAT, you see that QMAT is induced and GFP is produced. We can also see in the Western blot that our Cas9 was inducible based on the QMAT induction. So here we can confirm that we have a stable cell lines which expresses Cas9 system. Next, we ordered gRNA, which was targeting to exon 1 of a UPF3B system. And these gRNA is exactly at the proximality of exon 1. Apart from that, we also ordered gRNA that was not targeting any of the regions here. And then we transfected the gRNA, did the dilutions, and selected the clones. So most of these single clones, actually, we serial diluted and looked for the single clones, and these single clones were then expanded. And then this expansion was looked into the Western blot, and sequencing was performed. So if you see the Western blot, we got a number of clones. Here I'm showing two different clones, clone 1 and clone 3, which were completely devoid of any UPF3B protein. And when we sequenced this, we were able to find out the deletions in clone 1 about 55 base pair deletion, and in clone 3 we have 5 base pair deletion, and these leads into the complete knockout of the protein here. Now, we wanted to check whether this deletion of UPF3B leads into the hampering of NMD activity. For this, we tested a beta globin reporter system, which is well known in the NMD field. So beta globin leads into the generation of beta subunit in hemoglobin. And at the position number 39, there is a introduction of T instead of C, which leads into the UAG codon. Instead of CAG, it is UAG. And this kind of missense frame shift actually is seen in the beta thalassemia patients, and that is what is recapsulated, and a mini gene was prepared. So when these reporter system was transfected inside the cells, as you can see in the RT-PCR, the control cells were able to degrade PTC containing reporter system efficiently, whereas the knockout proteins where UPF3B was deleted, these transcript was still retained, right? Apart from that, we also tested number of endogenous canonical NMD substrate, namely Gas5, which is a long non-coding RNA, SRSF2, which is again known because it has a 3' UTR, Trojan exon is there, ZF-Fas, long non-coding again. So all these canonical NMD substrate were actually nicely degraded in control cells compared to the knockout cells. Suggesting that NMD is hampered when you knock out UPF3B cells. Now we also tested, as I mentioned, there are number of physiological transcripts which are also getting degraded with the help of NMD here. So we tested two different physiological level transcripts. One is GAT5 and another is GPX, both containing long 3' UTR, and these two transcripts were also upregulated compared to your control cells. We also tested its half-life. The half-life was increased from 7 to 9 hours in our extrapolated graphs. So we can fairly say that these transcripts are degraded by UPF3B. Now we wanted to actually find out whether these effects are directly by UPF3B or these are indirect effects. So for this, we took clone 1, which was devoid of UPF3B. We ectopically expressed UPF3B protein and then checked whether the phenotype can be rescued. And what you can see in both the transcript, there is a consistent actually restoration of degradation of GAT45 as well as GPX, claiming that these effects are regulated by UPF3B probably directly. Now to identify completely genome-wide this differential transcription expression was conducted with the help of DE-seq2. And since alternative splicing is the major contributor in NMD processes, therefore we also performed isoform switch analyzer to know what is the important event in alternative splicing which leads into the NMD processes and what is the consequence of that. So DE-seq2 actually gave us 119 upregulated transcripts which were commonly present in both clone 1 and clone 3. One of the example I'm showing it here is COMD2, which generally is involved in NF-kappa B signaling. And here this transcript is NMD sensitive because of the exon inclusion at the position number, this fifth here. So another 2 minutes. So apart from that, there are number of transcripts which we see that because of exon inclusion, we see NMD sensitive transcript compared to control cells. And now when talking about alternative splicing events, what we see that in both clones, it is either the alternative transcription termination start compared to the stop site, or it is intron gain compared to the intron loss. That is the main contributor at least in UPF3B knockout setting, that contributes for NMD sensitive transcripts. Now, one of the example here is splicing factor. And when you can see that in knockout cell, these transcripts are NMD-based transcripts are majorly utilized compared to normal transcripts, and these were also confirmed by RT-PCR. So I will come to the last slide. So here we also tested first knocked out UPF1 because being a central regulator, what is the contribution into NMD. So if you downregulate UPF1, most of the transcripts go up because these are all degraded basically by UPF1, which is the central player, as I said. So most of the canonical transcripts are also upregulated in this case. But we found number of unique transcripts which were only degraded by UPF3B but not UPF1. And here I can say that there is a subset of NMD sensitive transcripts that are only degraded by NMD factor, specific NMD factor. And these concept actually is fairly new, whereas I have some example in the literature. Here in this article, they show that in sarcomere, it is UPF3B and not UPF1 or 2, which actually is localized in Z disk, responsible for degradation of PTC containing myosin binding protein. Not only this, a very new article in May actually says that cell type and the factor specific NMDs are present in this case. In fact, last month there is an article which completely says that all NMD transcripts that are based on the EJC 50-55 based rule. And based on that, they have a huge open database that, that gives the example of NMD biotypes. So with this, I actually did a lot of separation between UPF1 and our clone UPF3B, and we found 64 novel transcripts. Most of these are coming from intron retention or termination stop codon. These are highly used in NMD process, and in the end, I thank all my team members. Most of the work was done by. Thank you for listening.
Thank you, Kusum. We have time for one quick question.
You said that it's probably UPF1, 3B is direct effect. How can you say that?
We did a lot of rescue, and most of these were again degraded, but we have to do all the IPs, RNA IPs. That is still
rescue
longer.
is just you're putting back.
Putting back, and of course, so these are one of the, because many NMD transcripts are not rescued. That is already known in the field. So this is a bit tricky.
Kusum.
Yes.
So generally NMD targets the shorter transcripts where this one intronic polynylation occurs. But what you find is that the longer UTR, which is also targeted by NMD. What do you mean by longer UTR? You find any features, or do you link with this APA? What do you mean by longer?
So longer UTR means where the stop codon, where you have poly binding protein, and the whole tail. The length actually increases, and that leads into NMD. But recently, all this sequencing assays says that it are shorter UTRs. Basically, that is fairly new.
Because UTR length is going to be determined by where the poly tails are added and those sites. So have you seen any link where the distal sites are used? And
this is already done in lot of reporter based construct assays. When the distance is longer, actually then they report.
Those are different in the
T3 assays. No, not in the cells. This has not been done.
Okay. In the interest of time, let's close the question and answer session. Thank you, Kusum.